Alright, in this video I'm going to show you how to use some of my computer models. In this case, I'm going to show you how to use the growth factors, cyclins, and cancer computer model. But let me take you to it. So to scroll to the different sections, starts off at the home page. You can just click to go to background information, and I've included in here a nice NPR article on cancer research. And Skittable by Nature Education has some good background reading information on the cell cycle. Under Explore the Model, here is a stripped down version of the basic computer model. I use Stella to build this computer model. You don't have to know anything about this, but it does help you see how the different interactions are taking place. For example, the G1 cyclin affects the S phase cyclin transcription. It increases it. Likewise, the M phase cyclin actually will cause a decrease in the G1 cyclin and the S phase cyclin. That's the basis of that computer model. Now, to actually run the simulations, there's lots of different features here, and I've included quite a few variables that you as a student can manipulate. For each, I tried to include information about each variable, and not too much, just a little bit. Okay, it sets you up with a default setting. So I'm going to run the computer model. If you just hit the play button, it'll run it. This will pause the computer model at any time frame. This will stop it, and if you would like to reset it, you can. Additionally, this is graphing right now the cell count. You can also go to a separate page which is graphing cell count again, the DNA per cell, which you can see is, DNA per cell increases and then it decreases, increases and then decreases. Likewise, M phase cyclin, anaphase, and so forth. So I've included actually with this model 5 different pages of graphs. You can use these different graphs to help inform on what's happening overall. Right here is the number of hours. So this is over 100 hours is where this is locating. So if you take a look at just the DNA per cell, and if I just scroll my mouse over any of the lines, different quantities at that time period will pop up. So I have 46 DNA molecules per cell here, and then at the peak it doubles. It goes to 92 DNA molecules per cell. And I can gain this information from my simulation. If I manipulate any one of these variables and I run it again, it'll run it with that new quantity. If you'd like to restore it to the default settings, you just hit undo and it'll restore it back to the default settings. Now I'm going to run the model again. I haven't changed anything over here, but you'll notice that there is going to be some variation here. And the reason there is some variation is I built variation into the model. This allows you to run your experiment, let's say 5 times, and then you could calculate things like the standard error of the mean, standard deviation, and so forth. Find the mean value of each of those experiments. But what I want to direct you towards, you'll see that variation right here. So after 1 cell division and 2 cell divisions, the first time we ran it, it was at 170 cells, and the second time we ran it, it's at 188 cells after the second cell division. And you'll notice at the end, hasn't quite finished running, but right at the end there was another cell division there. So it was up to 1,222 cells, and this one didn't go through that. So I don't think that would be a good comparison. So let's look here. It is 627 cells compared to 567. So while it does have kind of a doubling, it's not quite doubling each time. Let's go to the worksheet. So the worksheet is going to be a good way for you to follow along with the model. And the first question here I want to direct you towards is, what measured variable allows you to identify the stages of the cell cycle? So let's go back here. Now my other pages won't compare different runs. This is the run for just that experiment I did. And same with this one, and this one, and this one. But my first page, page 1, will compare my different runs, or my different experiments I've done. So this, while I could say, yeah, my entire cell cycle happened between here and here, and my entire cell cycle happened again between here and here, that wouldn't let me distinguish between G1 and G2, for example. So let's look at a different page here. And here you see anaphase, so that's when anaphase is taking place. Here, let's look at this one. So I have DNA per cell. Well, DNA is right here, it's staying the same, and then all of a sudden it increases. Well, what's causing it to increase is it's going through the S phase. DNA is replicating, going from 46 DNA molecules to the end of S phase of having 92 DNA molecules. Then for a very short time period, according to my model, it's in G2, preparing for mitosis. Then during mitosis, it splits that DNA up, and at the end of mitosis, it's back in half, where we have 46 DNA molecules again. Now we're in G1. G1, S, G2, G2, mitosis, G1, S, G2, mitosis. So that's how you could, that's the variable that you would look for, DNA per cell, to identify what stage of the cell cycle. Now my model is not perfect. Usually for a eukaryotic cell, it's going to be in the S phase for a rapidly dividing cell for like 6 hours or longer. But for my model, it's only there for, it looks like 1 hour. So that's one of the inaccuracies of the model that I have for you here. If we go back to the worksheet, we can also cause our cells to have cancer. And if we look at our simulation, we could actually, let's go back to that first page. So one of the hallmarks of cancer is the cell grows uncontrollably. So let's just increase this amount of mitogen. As we run this super fast, 2 rounds in 1 hour, and then it shoots up again at the end. And let's see, so here we ended up with 3,538 cells. Here we ended up with 1,222. So we have induced cancer into our cells because they're rapidly dividing cells. We could run this again, it will come up slightly different. I want to do something slightly interesting before I wrap up, is I'm going to increase this growth factor. Now growth factor should also increase the rate of mitosis and the rate of cells dividing and going through the cell cycle. But what you're going to see here is it doesn't. And this is built into the model, but I would imagine that you might see this in real living cells. What has happened is it's not going to end up growing much more, if maybe even less, than a normal cell. And that's because of the negative feedback loops that are built into the model. So let's take a look. Again, our cell count is pretty low. Well, if we look here, what has happened is this M phase cyclin keeps on decreasing. And let's look at some of our other cyclins. Let's see, where's a page of all? Okay, here's all the cyclins. Well, if we take a look here, our S phase cyclin is kept at 0. So why would our S-phase cyclin be at 0 here? Let's look at, here's all of our cyclins. Our G1 cyclin and our S-phase cyclin are both at 0 the entire time after that first round of cell division. Let's go up to explore the model, take a look. S-phase cyclin and M-phase cyclin and G1 cyclin. Well, your M-phase cyclin is actually causing a negative feedback. So this is really high. Your M-phase cyclin is really high. It's causing your S-phase cyclin and G1 cyclin to be low. And even though we tried to induce cancer by running it with lots of growth factor, lots of mitogens, it actually caused a negative feedback, preventing it from going through the cell cycle. So that's kind of how I use my computer models, and I hope you experiment with them and enjoy them. And any feedback, let me know. Thanks.